and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 173 for the podcast. Last week saw more than 5,900 people from more than 3,200 companies gather in Vienna, Austria for the continent's largest biopharma partnering conference, BioEurope. Over 3 days, the world's top pharma companies, most innovative biotechs, and life science professionals from 60 countries took more than 32,000 one-on-one meetings, made the connections, and sealed deals that will drive the industry forward into 2026 and beyond. Among the highlights of BioEurope this year was the Startup Spotlight Contest. This exciting competition offers biotech entrepreneurs an affordable opportunity to engage with top-tier investors and other important players in the biotech industry and to take their business to the next level. 8 finalists made their pitches at BioEurope, and one, Fusix Biotech from Germany, came out on top. This week on Beyond Biotech, I'm joined by Fusix Biotech's CEO and co-founder, Jennifer Altomonte, who tells me about her journey through the industry, Fusix's InFUSE platform, and what it's like to pitch and win one of the most important biopharma startup competitions of the year. I hope you enjoy my conversation with Jennifer Altomonte. Jennifer, welcome to Beyond Biotech.
Hi, Dylan. Thank you so much. I'm happy to be here.
Hey, Jennifer, just a couple of days ago now, Fusix Biotech was named the winner of the Startup Spotlight at BioEurope in Vienna. Congratulations. Your team must be over the moon right now.
Yeah, thank you so much. It was, I'm sure everybody always says it, but it was truly a surprise because all of the teams were so good, and I was actually not going prepared to win. It was more just the honor of being able to pitch and be seen, which was my aim. So it was super cool to actually win.
Jennifer, how did you come to biotech in the first place? I mean, what's the story there?
Yeah, I wish I could say that I always dreamed of going into biotech, but actually it's much more boring, I guess. So I've been working in academia for 20 years or so, doing translational research, but never really having any idea about how to make that translational leap until we had developed this oncolytic virus platform that started to show so much potential in being very safe and also very effective. And then I suddenly had to figure out how I could bring this to patients, because that was ultimately what I wanted to do. And I quickly discovered that to develop a drug within academia was not really an option because of the cost intensiveness of it all, and all of the bureaucracy involved in trying to do that through academia. And so I kind of figured out I could either out-license the technology to a company, or I could start my own company. And actually, at first, I didn't really think that the second option was really a realistic one, because I had no background in business. I had no idea how to found a company or anything about developing a drug. But I somehow just started informing myself about what would be involved. I started reaching out within my network to other people who had founded companies and to our university support. So the Technical University of Munich, where I work, is quite well known for entrepreneurship, and they have a lot of support available. And so they kind of gave me a few tips about like boot camps to go to, and non-dilutive funding that I could apply for. And that was kind of my first step in seeing how I could do this and getting some validation that this would really be an option. And it became a lot of fun to learn the business side of things. And I realized that to out-license the technology to a company would feel a bit like giving my baby up for adoption in a way, because I was so attached to it and so excited to see where it would go, that I really wanted to be a part of the process. And after I got the first non-dilutive grants to support the startup, I realized probably we had a chance, and that was how it got started. So it wasn't really something that I actively looked for. It was kind of just something that happened along the way.
Yeah, you say that we got going and we got going because you
Oh,
co-founded Fusix with a couple of different experts there as well, three other people.
yes. Yeah, but it's
How
kind
did
of
you
funny.
guys?
I think it's something something
How
you
did you
learn
get
in
together?
academia that you always say we. So the first other person was kind of just a no-brainer. She was a PhD student in my lab at the time and had um, expressed an interest in entrepreneurship. This was Teresa Krabbe, and she sort of was just with me from the beginning. And so it was a very natural thing that she would join me in the company. She was very active in applying for those grants in the beginning and helping me to make the first pitch and so on. The other two co-founders I sort of picked up also rather organically. Marcus Gerhard was a professor at our university, and I knew him from research, and I also knew that he had already founded a company prior, and I kind of reached out to him to get some advice and find out how he had done it, and to get some feedback about whether he thought that this could work. And he kind of took on like a mentoring role in the beginning and was giving me a lot of advice. And somehow this became more hands-on, and we both kind of decided that we would like for him to also be a co-founder. And then he introduced me to the fourth co-founder, Marianne Wiegand, who happened to be working in the same or on the same floor with Marcus and had extensive experience in viral vector manufacturing, which is an important aspect of our whole program because our drug product is a viral vector. And Marianne had worked in biotech already for quite some years and had kind of a different skill set to bring to the table. And so I started talking with him. He was very interested in the project, and personality-wise, it really fit very well. And so we brought him in as the fourth co-founder. And then together we founded in 2022. So it's been like 3 years now.
How did you divide up those initial roles in the company between the four of you?
It was kind of, in a way, obvious based on our background, right? So, I mean, I have the scientific background as well as Teresa. So we were kind of responsible for the scientific program. And then since I was leading the whole thing, I kind of fell into the CEO role. So I do most of the fundraising and business development. Marcus has a medical background, so he from the beginning sort of fit into this medical director role. So he's mainly responsible for the clinical development plan and communicating with the regulatory consultants that we work with. And Marian, because of his background, is now responsible for our CMC program and also serving as our CTO because he also has this very technical background.
I mean, as a scientist, and working in academia as well, you're always having to communicate data, communicate ideas. But the ideas that you're communicating when you're talking to a regulator or to an investor or to a potential partner, this can be very different. Have you had to make a transition there when you're talking to an investor versus a student or a colleague?
Yeah, definitely. I think this was one of the biggest transitions to make when moving from academia to a biotech startup because nobody really ever teaches you how to present scientific concepts in a non-scientific way. In academia, you're kind of trained to speak as technically as possible, and here we had to make a complete switch and sort of distill things down to very simple terms that non-scientists could also understand. So I think that was probably the biggest thing that I needed to learn. I mean, all of the business stuff, you kind of pick it up along the way, but being able to present effectively to a non-technical audience was a big switch. And then among the different partners within biotech, there's also a different language, right? So when you're speaking with regulatory consultants or the regulatory agencies versus investors versus pharma partners, you kind of have to speak their language and learn their kind of lingo. And yeah, that was also interesting to pick up. But it makes you sound way more intelligent if you use the proper terms. So it's something you have to pay attention to and put some effort into.
Let's talk a little bit about the science there at Fusix now. You've got a platform, it's called InFUSE, and it combines this fusogenic oncolytic viruses with a systemic IV delivery.
Yes.
There are lots of platforms out there, but there've always been historically a couple of limitations on the oncolytic viruses. And the three that I have here are the things like limited replication in solid tumors, weak immune activation, poor tolerability after you administer it via the IV. I mean, how are you working against this?
Yeah, basically we accomplished all three of those things through our fusogenic mode of action. And I will explain what that means because I realize that's a bit vague. So by fusogenic, I mean that our virus is literally expressing a fusion protein, and it's causing infected tumor cells to fuse with surrounding tumor cells. And this is essentially a chain reaction. So you can infect one tumor cell, and this leads to the infection of hundreds of tumor cells. And so in this way, we addressed the first limitation, which was limited replication in solid tumors. So this is kind of obvious, right? Like if the virus is really melting the tumor cells together, you get a much more amplified effect because of this chain reaction. But the other two things, sort of non-obvious, are also addressed through this mode of action, because it turns out that when tumor cells die through fusion, it's not a typical mode of cell death, and this is really activating the immune system through this process of an immunogenic cell death. So it's leading to the secretion of cytokines and these danger signals that then attracts immune cells to the tumor microenvironment and allows them to get activated. And this really reverses the whole immune suppression within the tumor, which is one of the biggest limitations of other modes of immunotherapy. And then there's the IV administration. And this is not working with other oncolytic viruses for several reasons. One of the big reasons is because most of those viral platforms are based on natural human pathogens that most of us have been exposed to before, which means that we have pre-existing neutralizing antibodies circulating in our blood. So if you inject these viruses intravenously, they will be neutralized by these antibodies, and then they're not efficiently delivered to the tumor because they're already inactivated by the time they reach the tumor. We overcome that because, first of all, our viral backbone is constructed using viral components from non-human pathogens. So these are veterinary viruses that don't naturally cause human disease. Most humans have never heard of these viruses or been exposed to these viruses. But the other thing is that because of this very efficient mode of action through cell fusion, if we inject the virus intravenously, even if it's not neutralized, it will still get taken up non-specifically. It will be filtered out by the liver and the spleen. And so very little virus actually reaches the tumor. But because of this chain reaction, we only need a few virions actually reaching the tumor in order to achieve a very potent therapeutic effect. So essentially, the answer to all of those questions is fusion. In my opinion, it's a very efficient mode of action, and counterintuitively, it also contributes to safety because it's mainly spreading from cell to cell directly. There's very little virus being produced or released into the circulation, and so we can achieve an excellent safety profile at the same time as achieving a very potent direct tumor cell killing and immune activation. So all of the boxes are ticked, and it can be injected intravenously.
Yeah, I mean, when I go to your website, you really emphasize three things, three pillars, really: the safety, efficacy, and delivery. Mm-hmm. Which of these three have you been concentrating on so much in these first couple of years of the business?
I would say all three. I mean, they kind of go hand in hand. You don't want to have a very safe therapy which doesn't work, right? And vice versa. Um, and I think the beauty of our platform is that it does all three of these things quite efficiently without much effort, right? So we additionally arm our vector platform with different therapeutic genes that can enhance the efficacy, but safety is something that's already there. So we've already demonstrated that the vector can only replicate in tumor cells. We don't see any replication or cytotoxicity in healthy cells. We've done biodistribution studies and a dose escalation study. We've not reached a toxic dose. So I feel like that's kind of already quite good. Of course, this will need to be tested in humans, but as far as we can test it preclinically, it does look quite safe. And then the efficacy is also quite good, and we can further improve that through expression of different transgenes and combinations with other therapies. And then delivery, we also feel that we've achieved with our IV delivery, although we're also always interested in novel formulations or other delivery routes. Depending on the tumor indication, we may explore more local delivery of the virus.
So your preclinical, is there a solid tumor model that's showing the best or the strongest complete response rate right now?
Yeah, so we've kind of tentatively selected hepatocellular carcinoma or primary liver cancer as our first indication for patients. This is primarily data-driven, so we've done pretty exhaustive tumor cell line screenings and patient-derived organoid screenings in vitro. And we could show that HCC was particularly responsive to our platform, although in general, it is effective in most tumors or tumor types. And then also in our preclinical tumor models in vivo, we could also show excellent efficacy in HCC. But this is not to say that it won't also be effective in other tumor entities. We also have quite nice in vivo data in pancreatic cancer, for example. But there were several factors that went into our decision beyond just preclinical efficacy, but also thinking about market, medical need, and also trying to balance where we can make the most difference with the amount of risk involved, because there are certain tumor types that might be quite responsive, but there are already effective therapies available where you wouldn't want to have to face that kind of competition. And on the other end of the spectrum, there are very difficult to treat tumors like pancreatic cancer, where there's the potential to make a bigger impact, but there's also a higher risk. So we try to balance out those things, and this kind of led us to HCC as a first indication. But we do plan to do our phase 1 dose escalation as a basket in order to get first clinical data on other indications as well, and maybe this will drive our decision in a different direction in other clinical studies later on.
I think the business side of things always comes in there. And another part of that business, though, is the IP protection that you had. You spun out of, or you took ideas that you'd been working on in academia. Now you have them inside the company there. How much of what you're working on is covered by new patents as opposed to previous IP that you worked on in the academic setting?
So currently it's all coming out of the university, and this was a big theme for us, how to license the IPs from the university. And this was quite a long negotiation process. Going forward, of course, even though we are still based at the university, so we're now kind of in this gray area where new inventions probably will be claimed by the university. This is also why we're planning to set up labs belonging to Fusix so that we can do completely separate research and have IPs that belong to us from the start. But currently all of the IPs belong to the university. I'm one of the inventors, but we as Fusix need to in-license the IP.
Let's talk a little bit more business. Let's turn to the Bio Europe meeting and the Startup Spotlight. There were 30 companies that applied. There were 8 finalists who made it onto the main stage. And of course, there was just yourselves as the winner. If I can actually take you back to the beginning, though, what prompted Fusix to enter the Startup Spotlight specifically this year?
Well, I'm not sure if I was aware of the Startup Spotlight in previous years, or maybe I had missed the deadline, but I wanted to apply just because I tend to think if you're eligible and you have the potential to either, win something cool or get visibility, then I do it. It's always a balance of time versus reward. But in this case, it was a pretty low bar to apply. And I thought, why not? Why not apply? Worst thing that happens is you don't get accepted, but that's kind of what you have to get used to in biotech. There's a lot of disappointments.
You had just 8 minutes and a little bit of Q&A in front of a big room of investors and partnering scouts. I was there, Labiotech was there. With all that pressure, how long did you actually spend putting that presentation together, testing it out, making adjustments with the team?
You kind of make me feel bad because, to be honest, I didn't really put in a lot of preparation. But I have to say, I've been pitching a lot over the years, and more recently, there's always a bit of a difference in the time allowance. So this is something that you always have to adjust for, but the content is always more or less the same. And actually, lately, I had been doing even shorter pitches, like 5 minutes or 6 minutes. So this 8-minute pitch was it 8 minutes or was it 6 minutes? Now I'm confused.
It might have been 8 minutes, 8 minutes total, including a couple of minutes of Q&A. Yeah.
Yeah. Okay. So, I mean, this was quite luxurious for me. So it actually allowed me to slow down and relax. And so, I find that a lot easier than these very short pitches. But I don't know. I'm almost embarrassed to say I didn't really put in a lot of preparation. I had my deck. We had to submit the deck, I think, 1 week before or a few days before. So that was already done. And then I just kind of went through it in my head a couple times on the day. And that was it. And I think for me, that's kind of the best way. I'm not someone who does well by over preparing.
The jury there got to quiz you at the end. Was there anything that surprised you in the Q&A or anything that you hadn't heard before?
No, I quite liked the questions. They allowed me to expand a little bit more on what I would have liked to say had I had more time. So actually, the questions were quite nice. I think they were mainly focused on our payload. So the choice of a soluble PD-1 as an immune checkpoint inhibiting payload. One comment that did surprise me, though, was that they would have liked to have seen more data in the pitch, because in such a short pitch, usually you don't want to show a lot of data because you just lose half the audience and there's not really enough time to explain it sufficiently. So I was a little bit surprised by that. But in the end, I guess they thought it was enough data to convince them.
Yeah, more than enough. You guys were the winners there. You were also the first to pitch. So you pitched and then you sat through all of the other presentations, waiting there. When they finally announced the winner, how did you feel?
I was completely shocked. Like, I was not prepared to win at all. So I wasn't nervous. Actually, probably that was helpful, because I had obviously looked up the other teams beforehand. I looked at their homepages and what they were doing, and I felt like, wow, they're all quite good. Some of the companies were even a bit more advanced. I think 2 of them are already at the clinical stage, and so I thought, okay, I don't really have much, to compete with them. So I was pretty relaxed, and it's also quite a luxury to be able to go first, because you don't, normally when you're sitting through other pitches, then you're starting to get nervous because you're comparing yourself. And in that case, I was, fresh and could get it over with, and then I could just sit back and really pay attention and enjoy the other pitches, because I didn't have to think about what I needed to say.
Talk about the interest since then? Has anyone reached out following the competition? I've seen you on LinkedIn. I've seen you in emails that BioEurope has sent out. Has there been any interest in connecting with Fusix?
Yeah, actually, I've gotten a lot of connection requests, a lot of emails. It's always hard to see where it will go. Usually, it takes a while before you start to see whether there was any real value in these connections. But I think expanding the network is always good, and visibility is always good as well. I like to keep on people's feeds to keep seeing our name, and it starts like oftentimes people will say, "Oh yeah, you guys are making a lot of momentum. I keep seeing you on LinkedIn," and I always have to chuckle because it's usually these small little wins or something that just makes people gives the impression that we're doing maybe more than people would appreciate if you were not so visible. So yeah, it's been good. We will see where it leads. We're fundraising right now, so I'm hoping
well,
we
that's
might make
that's what I was
some
going to
new
ask about. Yeah, because I mean, you're chasing money. I mean, this is obviously of interest potentially to pharma, but also to venture capital who might be investing. Are these the sorts of connections that you want to make right now? It's all about fundraising?
Absolutely. I mean, fundraising as well as strategic partnerships, although my highest priority right now is really fundraising. I think that's the first step, because until now we've had only non-dilutive funding, which has been great, but we can only get so far with non-dilutive funding. And I feel like the timing is right now in terms of our development, that we really get a larger amount of funding in so that we can really focus on manufacturing scale up and doing all of the IND enabling toxicology studies, as well as setting up our labs and getting ourselves more operational than we have been. I think this will be crucial in really scaling ourselves as a company. So that's my first priority. But of course, business development is going to be crucial in going forward, and especially for bringing our assets to the market, because this is not something that we will likely be able to do alone as a small biotech company.
For other small biotechs, and particularly in the oncology sector there, based on your experience in the last week, is this something you'd recommend that other startups get involved in, this startup spotlight, doing that pitch, getting up in front of people in public and making that big call?
Absolutely. It's not only great for meeting people, expanding the network, getting visibility, but it's also somehow really good practice, and it helps you to really sharpen the pitch, and it prepares you for those investor meetings. If you're already up there pitching regularly, it becomes just kind of your second language, and you at some point it's just very natural. And so if nothing else, it helps to get better. So in the beginning, as I said, I was not at all familiar with how to pitch as a biotech company compared to as a postdoc. And when I think back of those early pitches and even some of those events that I went to that I clearly was not ready for, it was all great practice, and it brought me forward. And so I think it's never really too early to get out there, especially if it's something that you have to apply for. There's a screening process anyway, so you probably will not be put out there if you're completely unprepared.
Jennifer, let's talk about what happens next now. Looking at your roadmap for next year, what's the biggest milestone that we should be watching out for?
So actually, what we're preparing for right now is to do a tech transfer to CDMO and doing those first scale-up steps. And this is, it maybe sounds trivial, but manufacturing viral vectors is definitely not, easy or something that is just an off-the-shelf process. It's one of the bigger risks actually in our program, and we've already developed these processes on a smaller scale. We've scaled it up to a point, but we can't really scale up further in-house. So now we're preparing to do a tech transfer and scale up to the full process. And this will be a big de-risking step for us, and it will allow us to come closer to clinical development, because it's one of those dependent processes that, obviously we can't apply for a clinical trial until we have a GMP product, which we've also tested. So in the next year, I hope that we will be able to demonstrate that we can produce enough for a clinical trial. And then the plan is by end of 2027 that we can dose first patients.
So what's a successful 2026 look like then for Fusix? Is it doing that tech transfer right? Is it raising some money?
Well, I would be most happy if we raise money, to be honest, because everything else is dependent on that. We can do the first scale-up steps with what we have. We just got some non-dilutive funding for that. But it will be crucial now to raise VC funding that we can really ensure that we can get to our first clinical trial. So that's what I'm hoping for. Everything else is just icing on the cake.
Jennifer, it's congratulations again for the success up there in Vienna. Where should listeners go for updates on Fusix and updates on the work that you're doing?
Yeah, well, first of all, thank you so much also for having me. For updates, best place to go is actually LinkedIn, because there you get the most current information. But you can also go to our homepage. Maybe we can link that in the...
Absolutely.
In this, yeah. There you can find out more about our technology and our pipeline, and for updates on what we're doing and what we're up to and our latest milestones that you will find on LinkedIn.
Perfect. We're going to drop in the link to Fusix Biotech in the show notes. We'll link as well to the Startup Spotlight and our friends at EBD who organize Bio Europe. Jennifer, thanks for being on Beyond Biotech today.
Thank you so much.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.